today's session, which is on the topic COVID-19 vaccine at the end of the tunnel. On behalf of Global Marketing Services Pakistan, resource group of companies, and our partner Chughtai Labs Pakistan, I welcome our high-profile speakers, scholars, and scientists who are attending this webinar from different parts of the world and across the country, Pakistan. First of all, I am thankful for support extended by Canada-Pakistan Research and Development Council, National Institute of Health Pakistan, and Drug Regulatory Authority Pakistan for accepting our invitation and extending active support to make this event successfully today. Global Marketing Services Pakistan took initiative to combat the COVID-19 when it arrived Pakistan in Feb 2020. And we supported nationally different departments like NDMA, AFIP, NIH, private labs like Chughtai Lab, Shaukat, and others by providing quality molecular testing and supplying timely with our services and supports across the country. I'm pleased to share that GMS Pakistan is also launching Illumina NGS into Pakistan, and I hope this technology will help our scientists and scholars to get more awareness about the mutant strains and for the better scientific research. We are here and taking initiatives by providing a platform where our scholars, our scientists can discuss to find a better option and solution among all the available options for vaccines. And we are taking this initiative and bringing our scholars and our speakers and our scientists to discuss today, and then of course to see the available options with the current challenges, especially after the emerging of the mutant strains. I trust we will have a better understanding after the enlightenment of the lectures and discussions. And without wasting the time, I will pass on to Major General Amir Ikram, who will take the proceedings to the speakers and he will proceed. Thank you very much for everyone.
Bismillah ar-Rahman ar-Rahim. Thank you so much, Mr. Zafar. Extremely grateful to Global Services for making it happen once again, and extremely delighted to welcome all the dear experts and also a good number of participants to COVID-19 vaccine light to the end of tunnel symposium. I'm sure we COVID understand truly that it's a big challenge, but such challenges do bring in opportunities, and it's our job to pick up those challenges and convert them into opportunities and do something. I think for the first time in a country like Pakistan even, we have been very successful in picking up all those challenges, picking up those opportunities. And now as we talk of vaccine, but side by side, as we move on with the vaccine, we do hear about different strains and efficacy of all that. So let's hear the experts. And I'm extremely grateful to all the experts for making it possible. Some of the rules of the games today would be that time has already been deputed to each expert. And we will move on, start one by one. I'll keep introducing them and inviting them. My humble request, at that time, you must put on your video as well, so that the participants can see those learned faces as well. And restrict yourself to the designated time. And finally, moving on, we'll be having question and answer session at the end. So once again, I'm extremely grateful to all our dear colleagues from GMS, Canada, Dr. Zair especially for making all the efforts to making it possible, and let the game begin. So first of all, I'm going to invite Dr. Mohammed Hamid Akhtar, and he's basically an assistant professor at the Department of Pediatrics at University of Toronto. I have had an opportunity of meeting him, a very capable person, and after completing his medical from Dow, Karachi, basically his residency training, UK later on, he moved to Canada, and he's Director of Pediatrics and Director Resident Education right now. Has been affiliated with University of Toronto and York University. So we start with your message, please. Over to you. Thank you.
Thank you, Professor Amekram. Bismillah ar-Rahman ar-Rahim. Dear speakers, participants, and attendees, Assalamu alaikum. As the President of Canada-Pakistan Research and Development Council, it is an honor to be here at the international webinar. I'm a pediatrician by trade and a faculty at the University of Toronto. The Canada-Pakistan Research and Development Council is a non-governmental organization based in Toronto. And its offices are also in Karachi and Islamabad. This organization focuses research through an exceptional, efficient, and formal affiliation between Canada and Pakistani institution. CPRDC has been actively working towards its goal since its conception, addressing United Nations Sustainable Development Goals, such as health and well-being, quality education, industry innovation, and infrastructure. CPRDC began with a simple desire to realize scientific development opportunities between Canada and Pakistan. After much hard work from Pakistani descent Canadian professionals, we have been able to create fruitful connections and have helped establish, facilitate, and negotiate interaction between the scientific institution in both countries. The Council has embarked on several projects and has signed memorandum of understandings with reputable scientific institutions, such as in education, Brock University and University of Karachi. As in clinical trials, Manitoba University and the Indus Hospital, Dow University of Health Sciences, Mayo Hospital, and National Institute of Health. Also, CPRDC provided opportunities for education for Pakistanis by creating positions of doctoral and postdoctoral candidates and the University of Toronto. We strive towards acting as a catalyst in research endeavors through Canada and Pakistan. Ministry of Foreign Affairs recently launches as science diplomacy. I would like all of you to visit that portal, and I'm sure it will be of some benefit. We have now a few focus groups working independently under the umbrella of CPRDC, and their members are from Canada as well as from Pakistan. Our focus groups include life sciences, health sciences, engineering, fintech, and innovation and digital sciences. I'm happy to see so many people with strong ambition and high achievements, and gladly welcome your support in our initiatives. I would like to say here at the end, thank you for the members and the team who has helped to organize this webinar, and special thanks to Dr. Zair as well. Thank you.
Special thanks to Dr. Akhtar. What CPRDC is doing, really, it's a remarkable job, and it's time to strengthen further and nurturing relations with Pakistan. Thank you so much. We move on to our next speaker. We have the privilege that we have the Consul General from Canada, and this talks volumes about the passion of Foreign Office and people over there putting their efforts toward the scientific side. And recently, the Ministry of Foreign Affairs started off the science diplomacy concept. I think this will pave the way for much better clinical and medical research in Pakistan. Mr. Abdul Hamid is one of the driving forces definitely behind all these ventures. He's basically a graduate of the Fletcher School of Law and Diplomacy, and has worked in various capacities in various countries, including United Nations. He has been a research associate in the Institute of Diplomacy at Georgetown University, and he joined in January 2020, right before the COVID crisis. So over to you, Mr. Abdul Hamid. Thank you so much.
Thank you very much for this timely seminar. In Toronto, we have been brainstorming various issues relating to COVID-19 under the Science Diplomacy Initiative and CPRDC during the last few months. I shall not focus on activities of CPRDC because Dr. Akhtar Hamid has already highlighted them. My conversation of today will be based on general observations that we make here, and as a layman, I shall highlight only on one broad long-term issue that requires attention. Sometimes our discussion on the COVID-19 ends at the gloomy conclusion that after this, there will be another epidemic. Our planners should prepare for the next pandemic. It's not a matter of if, but when. High growth rate of our population, and as one of the most affected countries in terms of the changing climate, we need to prepare ourselves and strengthen our national health system in general, and vaccine and medicine manufacturing capacity in particular. There's need for developing core capabilities to deal with future epidemics in Pakistan. We need the long-term manufacturing capacity of vaccines and medicine at national level, and investment in the infrastructure and research and development as a prerequisite for it. This proposal should not be ignored with the easy to say, hard to do rebuff, because it is both strategic and developmental imperative for a country that has the 5th largest population in the world. In addition, we also need the adequate number of trained health workers, effective health information system, including surveillance and public financing that meet the requirements for implementing these proposals. In this age of pandemics and international forums, the conversation starts like this, that no one is safe until and unless everyone is safe. So it highlights the need for international collaboration. No country alone can protect itself from pandemics. Solidarity and international cooperation are vital. Having made this liberal point in terms of international theory about multilateral cooperation, one needs to be a realist as well. At the cost of sounding like a vaccinationalist, one must state that it clearly that meaningful and long-term international collaboration will follow and not precede our national capacity. The initial focus should be on creating required capacity to deal with epidemics at national level. The light at the end of tunnel for me is not that we shall somehow manage to get vaccine for this epidemic. The light at the end of tunnel is that COVID-19 has acted as a wake-up call for Pakistan, and hopefully we shall face the next epidemic with our national capacity in place. Some experts in Toronto believe that if Pakistan attaches due importance to deal with future epidemics, this kind of permanent capacity based on indigenous or collaborative research and development can be created within the next 3 to 4 years. It is not too ambitious an idea for a nuclear state like Pakistan. It is doable. Many countries which started their journey of development with us have done it. Pakistani expatriate communities' expertise around the world could be tapped for it. However, the basic initiative must be taken within the country. I leave it to experts whether a separate institution needs to be established for it on the priority basis, or it can be done within the existing institutional framework. Vaccine development is not only a health issue these days, it has far broader implications for countries' economies, strategic advantages and disadvantages, security, as well as diplomacy. Pakistan has no option other than developing such a capacity to manufacture vaccines and medicines at the national level before the next pandemic hits us. Thank you very much.
Thank you so much, Mr. Abdullah. You're very right, truly eye-opening, and already a lot of hard work has been done in this regard, and surely we would like to share shortly with your kindness as well how we would like to move on and how we have strategized. So thank you so much. Now I would like to invite a man or a man within a man, so many things, so many faces and all so elaborated, so it is very difficult for me to define him. Dr. Bhutta, Dr. Zulfikar A. Bhutta is basically a Robert Harding Inaugural Chair in Global Child Health at the Hospital for Sick Children, Toronto. He's a co-director of Sick Kids and founding director of Center of Excellence in Women and Child Health, Aga Khan University. He also holds adjunct professorships from multiple in universities like Johns Hopkins, Tufts, University of Alberta, London School of Hygiene and Tropical Medicine. He has been designated as National Professor of the Government of Pakistan and among the founding members of Ethics Committee. He's been part of member independent expert review group appointed by the UN Secretary General for monitoring global progress in maternal and child health millennium development goals. He represents the Global Academic and Research Organization on Gavi Alliance Board and is co-chair of Maternal and Child Health. And there's so much, frankly speaking, I can continue, but I would like to hand over to Bhutta. Dr. Bhutta, over to you. Thank you so much for joining us.
Well, thank you very much, Amir, for that generous introduction. You forgot one thing. I'm a close friend of yours, and that of national institutions like the one that you're heading at the moment. I actually want to build on what Hamid Saab just said, because I think it's important, and I am having a quick look to see if this is largely a meeting of national experts, and there are about 130 attendees who cannot see, so I have to measure my words very carefully. As some of you know, particularly Uzair, an effort was made about 20 years ago to create national capacity in vaccine development. At the time, the prerogative for that arose, and I chaired that task force appointed in Musharraf's government and through Dr. Ataur Rahman for several years. The prerogative arose because it was very evident after 9/11 and the consequent aftermath of that, that in some areas, doors would close to countries for a variety of geopolitical reasons. One of those was because of the nature of our engagement in that particular field was really particle physics at a higher level. But the second that was very apparent after the 2001 was also research that was at the cutting edge of viral genomics, microbial pathogenesis, largely because of fears of dual use and the risk of those technologies falling in the hands of quotes unquote other states. And that's one of the reasons why we did not have progression from BSL 2s to 3s to 4 labs in the country with the capacity for doing that kind of work, which we recognized at that time was a national security imperative. And as Hamid has said, these are investments that you have to make with a long-term vision. So we tried, and we tried very hard. And as General Amir knows, at that time, the imperative was to have the public sector lead it. The NIH at that stage had some capacity with General Burney, who was so very kind to work with me at that time, felt could be expanded upon, and to bring the private sector in as well. But to cut a long story short, the effort had to be abandoned after 2 years, not for reasons of a strategic planning process, but because of a range of views, including one of which was that this is a wasted investment because we will always get the vaccines that we need. Why? Because we had joined Gavi and we were assured a supply through Gavi, and also that you could get it cheaper in the open market. Well, there are some things you cannot buy on the open market. And one thing that was not recognized at that time, even though we put this forward as a national security imperative, was that it wasn't just vaccine development, it was all the other things that come with it on research and development, on immunology, on the biotechnology platforms, on the ability to make diagnostics at the drop of a hat. And this needed to be done systematically as a means of getting Pakistan to a stage where we were no longer dependent upon others for our support. And the idea was not just to do it for Pakistan as a nationalistic approach. it was also put forward as something that the entire Islamic world needed to do. And today, 20 years on, it saddens me to say that we sowed what we reaped. And we therefore ended up by trying to address the COVID-19 crisis in an emergency mode. And thanks to many of our national agencies and institutions that still have some depth, that we've been able to do many things at a level of competence that's been appreciated by the world. But that still leaves us very vulnerable. So it gives me absolutely zero pleasure at this stage to say, wow, you know, this weekend we'll have 500,000 doses of vaccines arrive through the courtesy of one of our closest allies. And then there will be some more donated. The bottom line is that we have, as a nuclear state, as a sovereign state of 20 million people, we've been forced to join a long line of countries who are dependent upon hand-me-downs. And as you can see with vaccine nationalism at this stage, countries are blatantly in utter disregard of the ethics of global equity and justice, placing themselves at the front of the queue and trying to enforce their sovereign right on vaccines just because they happen to have either supported their development or the production facilities lie in their region or territories. So to cut a long story short, I hope that at some stage, and we are taking some steps, as some of you know, either through the Higher Education Commission in discussing its research priorities, or on the 4th of February in a high-level roundtable discussing exactly these issues, the lessons and opportunities that arise from a review of Pakistan's ability to handle COVID-19. So we are trying to move forward on this, and our premise there would be that we need to bring together all capacities that we have within the country, but increasingly recognizing how little investment has taken in this space over the last 20 years in our universities and academic centers. I think this cannot be done without engaging the diaspora and some of our colleagues in the wider network across the world. My last point on this is that this will take time, effort, and this will also require sacrifice. And nations never build anything without prioritizing where they need to invest. When we needed to invest in some technologies in the country for our sovereignty, for our national interests, and for the fact that we couldn't buy those things off the market, we did not hesitate to make those investments, and at the highest level. And even today, it's not just our nuclear capacities, or capacities in many things. When we put our mind to it, we can do it. And Amir will tell you that when we needed to produce personal protective equipment, whether we needed to produce hand sanitizers, and even homemade ventilators, we were not found wanting. But I hope that this will also take some practical shape and not just rhetoric. And it will be followed by concerned agencies and authorities, both authorities with power and authorities with moral authority, in translating this into action. So I'm grateful to be part of this. I'm largely in a listening mode. I actually canceled something to be able to attend this, and I will look forward to learning from you as to how you plan to take things forward, not as users of technology or recipients of technology, but as perhaps developers of technology. Thank you.
Thank you so much, Dr. Bhutta. And let me share, inshallah, soon he will also be the part of new board of governors of NIH, and we are sure whatever we have already strategized for vaccine production, inshallah, under his able guidance, we would like to move on. So thank you so much to all the colleagues for remaining very much in time. As we move on, the next person, he always reminds me of a movie, A Few Good Men. And alhamdulillah, there are many good men in any case, but he's a person from Drug Regulatory Authority of Pakistan. And I am eyeing him as one of the futures for DRAP, inshallah. He's basically a pharmacist by profession with a PhD degree to his credit, and has been associated with DRAP since 2001. A man with definitely with good potentials, and I'm sure with his, I think, true sport, DRAP can also flourish in the near future, because it is mandatory to develop DRAP side by side with other than institute, so that we come up not only to the national expectation, but international as well. Over to you, Mr. Badullah.
Thank you very much, Professor Amerikam, and all the participants, and especially to the organizers, Professor Dr. Uzair, for putting all efforts to organize such a useful webinar where all stakeholders and professionals can share, and definitely they will share their expertise and knowledge. So topic of my presentation is whether it's not appearing. Ms. Sidra, would you like to share the screen? Exactly. So topic of my presentation is a regulatory landscape of COVID-19 vaccine. But before going directly to the COVID-19 vaccine, I would like to share a few slides which will definitely set pitch for further discussion. My next slide is about the WHO health system framework. And you can see that WHO has established health system on the 6 parameters and 6 component approach. And one of the foremost is the access to essential medicines. And if we see that how United States has set sustainable development goals, which have already been shared with Dr. Akhtar Hamid, and we can see that there are the 17 goals for the SDGs, and third goal is the good health. And every country is mandated to achieve this good health as a sustainable development goal. And one of the target is the availability and access to safe, effective, quality, and affordable essential medicine and vaccines for all. Now, in such a situation where there is a COVID, then this is very important and strategic to have all these medicines, and specifically vaccines, available to all people of the country as general. So if we see that now how we can achieve this all vaccines and affordable essential medicines, there must be some system, and definitely regulation ensures, a regulatory authority of any country ensure and assure to make available for all these things. If we see that a routine vaccine development timeline, then it is very much clear, and we all know that it always takes several years to develop a vaccine, starting from its preclinical studies, developmental studies, designs, and then process development, preclinical toxicological studies, and then comes the clinical trial, phase 1, 2, and 3, and definitely safety assessment and all the assessments which are related to the immunogenicity as well. So all these clinical trials, and after these trials, then definitely applicants submit the regulatory submission to the regulatory authority for the review and decision. And at the end of the regulatory decision, if it's the approval, then it's the large scale production and the distribution of that very vaccine, along with some safety studies or post-marketing studies. And in all such cases, it usually takes 4 to 5 years to develop such a vaccine. But if we see that in case of COVID, it just took 10 months to develop a vaccine in the market. Now, vaccine researchers have put all their resources for the development of vaccine quickly, and definitely it is only due to the unprecedented level of funding and global cooperation as well. We still remember that first vaccine which was appeared, that was after the some days after the coronavirus genome were made public, and at the same time regulatory processes started up, and then we can see that all the processes which were mandatory for the development of vaccine, that were sandwiched, and after some time
the vaccine was available in the market. And in such situation, it was altogether fast-tracked globally. Development is compressed in time, applying extensive knowledge on vaccine production, and then vaccine was available. And it was only due possible that there were parallel approaches for the studies, phase 1, phase 2, and phase 3. In some cases, that in early phase 3 trials, then there was a significant results. Some regulatory authority even permitted the use of that very vaccine. And you can see that the clinical trials were also overlapped. And at the same time, when the vaccine was in phase 3 trial, there was risk manufacturing and bulk manufacturing was also started. And in such a way, all the scientists work and regulatory authority work, and we have vaccine in absolute 10 months time, rather other than that. If we see, there are basically 5 types of vaccines which was by and large explored, and on the basis of these vaccines, now we have about 6 vaccines which are available in the market, and these various types of vaccines are DNA, RNA vaccines, live attenuated, inactivated, and another one is the viral vaccine. And subunit is still under process. And it is the snapshot of global vaccines which have been under consideration by the World Health Organization for its pre-qualification status. And we can see that by the end of 2 months further, we can have some WHO pre-qualified vaccines as well. So it was all about the global scenario of the vaccines, how the vaccines were developed, and how the various regulatory authorities worked for the approval of these very vaccines. If now coming to the national DRAP scenario, that after the existence of DRAP in 2012, the DRAP pledged itself for the adoption of global regulations and to adopt all the internationally harmonized legislations. And by and large, now we are the WHO Collaborative Registration Procedure member, we are the member of Uppsala Monitoring Center, and we have adopted common technical document, and so on. We have also signed some MOUs with various regulatory authorities. And taking advantages of all these things, now all these COVID-19 related vaccines were also evaluated on the basis of all these international harmonization and global agreements as well. On the basis of this, DRAP has shortlisted and entered into some sort of MOUs with various regulatory authorities. There are 19 regulatory authorities and 2 agencies to whom DRAP has pledged and signed various MOUs for the various recognitions. It's before you. And on the basis of that, all together, after the DRAP establishment, now we have switched over to the all those vaccines and all those products which are only approved by these reference regulatory authorities, and based on our reliance principle of the WHO and ICH. If we see that regulatory status of various COVID vaccine in Pakistan, then we can easily say that it is the registration board which is the statutory board for the grant of registration. But as I have said that right now, none of the vaccine which was approved by the reference regulatory authority, as already shared by Professor Bhutta, is not still approved by DRAP because we are not in their priority list. So we focused on some vaccines which was approved by other non-reference regulatory authorities. And now we definitely, in order to provide the vaccines to our country, then we shortlisted some vaccines, and right now we have approved 3 vaccines from the non-reference regulatory authorities. But this approval was not altogether. There is a long work which we have done on the basis of the expert working group which was constituted globally, and Professor Bhutta is also associated with that very working group. And all the clinical trial data were evaluated by this working group, and it's a 7-member working group which held series of meetings and reviewed clinical data. And right now, 3 vaccines have been granted emergency use authorization by DRAP, and these vaccines are Sinopharm vaccine, which is being manufactured Beijing Institute of Technology, biological products, Another one is the AstraZeneca vaccine, which is being manufactured by Serum Institute. And another vaccine is the Sputnik vaccine, which is the Gamaleya Russian vaccine. So hopefully the first consignment will be reached by next week, and we will be able to launch our vaccination campaign. And one thing very important, that is the race versus race time against performance. So for this purpose, for this all approval purpose, DRAP has worked very diligently, and within the days we have done all the evaluations, and our expert working group works on the daily basis, and the basis of that we approved all these 3 vaccines. And as the title of our seminar is light at the end of tunnel, so taking advantage, it's the first time in the history of Pakistan that such a type of clinical trials have been also conducted in here and approved by the DRAP as well. First and foremost is the phase 3 clinical trial, and about 18,000 subjects were recruited, and this trial is being done by the Professor Amir Ikram, being the principal investigator, and it's about the carcinobiological China, and it's one of the very modern clinical trial which has been approved by DRAP, and such a large trial was never conducted. And similarly, phase 1 trial of Beijing Sinopharm was also approved. So to summarize, like other parts of the regulatory parts of the world, DRAP has also worked for not only the approval of the vaccines, rather, and for the clinical trials approval. And as Professor Bhutta has also shared, that there is an intense need of the indigenous manufacturing of the vaccine, although we are working since last 2 decades, but still we are not able to do anything. But we do hope that with all these things, and being taking advantage of this COVID and light at the end of tunnel, we will be able to develop some sort of local indigenous production in our country as well. Thank you very much.
Thank you so much, Dr. Abad. I think, and we are witness, because even yesterday we had a meeting at the Parliament where DRAP was supposed to chart out the line of action for the near future. So things going parallel, alhamdulillah, that's the strength, I think, and as already we have the potential to move on. The next speaker online is Dr. Syed Irfan Asif, and just watched him some time back, and the person really fascinated me. And he's presently the senior consultant at Sanofi Pasteur, have had experience of quality and operational heading in various countries like UAE, Canada, Saudi Arabia, besides Pakistan. He holds a PhD and is an author of 4 books on the pharmaceutical side. So over to you, Mr. Irfan.
Thank you very much for this very kind introduction, Dr. Major Amir Ikram. Do I have to share the screen, or is it control over there? Okay. So why it is not showing up? Can you see my screen, or?
Yes, we can.
Yes, we can.
Yeah. Okay. All right. Okay, so here we are talking about the industrial perspective of COVID-19 vaccine, talk of the town these days. And we are going to take a look of the variables, advantages, and challenges that the industries can meet or can face to manufacture these vaccines. What an ideal vaccine could be? Obviously, I mean, no side effects, same immune effects, not causing other diseases, long-lasting protection against the virus, and prevents further spread of the organisms through the vaccinated person. And so far, the clinical trials have shown that the newly developed vaccines that we have been talking about, they have many of these. long-term ongoing surveillance of the disease.
Sir?
Hello? Is somebody saying something?
चलिए अगर स्पुतनिक वाली वैक्सीन का पूछें तो चलिए ठीक है सर
ठीक
Is there somebody talking or something?
No, sorry, you can move on.
Oh, okay. All right. So long-term ongoing surveillance of the disease, it will show further whether, the vaccine protection is long-lasting or booster annual doses are needed or not, or the vaccines prevents a vaccinated person from carrying and spreading the virus. I have noticed that Dr. Badullah has just highlighted the types of vaccines that we are currently talking about. And so I'm just going to skip this slide. I'm not going to touch a lot on this for sure. He has already mentioned about the different types of vaccines, the mRNA based, which are the first and foremost of the the vaccine type. A lot has been spoken about this the type, which is very exciting. But they're definitely fast development speed, low to medium manufacturing scale, and of course, we all know that Pfizer and BioNTech are manufacturing these vaccines. DNA type of vaccines are also not entirely new either, and a lot has been worked on these type of vaccines, and I think they have been used in gene and cancer therapy. Protein subunit, purified pieces of pathogens, they're medium to fast development speed, high manufacturing scale, 2 doses required, Novavax is manufacturer. And then the whole virus, the traditional type of vaccines, fast development speed, which is being manufactured by Sinopharm and Sinovac, also needs 2 doses. So next slide is about the advantages. What are the advantages of these vaccines? They possess different advantages and disadvantages as well. So we see that the mRNA and DNA, well, no question about the safety of these type of vaccines. As there is no live component is injected in mRNA-based vaccines. And for non-replicating viral vector, we see that also very well established technology that can trigger the strong immune response, as it also involves both B cells and T cells. The protein subunit, well established advantages for those having compromised immune systems. And similarly, the whole virus, the conventional vaccines, the technology is of course well established, so suitable for people with compromised immune system, and it is relatively simple to manufacture. Challenges. So the mRNA, the first one, mRNA based, we have seen a lot of talk about these type of vaccines. It's entirely a new type of vaccines, making people skeptical about using it, and but ensuring the effective delivery into the body, since free mRNA is it tends to quickly breaking down, and of course the storage issues with the Pfizer and Moderna vaccines, -70, -20 degrees centigrade. So challenges with the non-replicating viral vector vaccines, previous exposure to the vector could reduce effectiveness. These type of vaccines are relatively complex to manufacture compared to others. Protein subunit type, relatively complex to manufacture. Adjuvants and booster shot may be required. And then whole virus, booster shot may require. So as a conclusion, we see that the cold chain supply in the world today is proving capable of and are successfully meeting the challenges for storage and transportation of Pfizer, BioNTech, and Moderna vaccines. Scientists and experts at various levels are educating people about the new type, this mRNA-based vaccines, telling people that it is safe and there is no problem using them, and it's a lot of myths actually going around, and they're trying their level best to make people sure that this is safe. Manufacturing on large scales to provide vaccines to the entire world population is also a bigger change. It's not an easy target. However, industries are working at their fullest capacity to meet up this challenge as well. In a nutshell, the world finds some light at the end of the tunnel, and the year 2021 definitely has got a better start than the previous year. As Dr. Abdullah also highlighted in Pakistan, what part Pakistan has played in clinical trials, we can see that there are many industries in Pakistan having state-of-the-art facility and are capable of making these vaccines instead of relying on the import from other countries. So we all hope for the best for the country and the people living in this country. I end up my presentation here. Thank you very much for your time. Dr. General Amir, you are mute. Please unmute your.
Oh, sorry.
Yeah.
Oh, sorry. Can you hear me now? All right.
Yeah.
Sorry. So I would like to introduce now Dr. Hamid Merchant, another fascinating young Pakistani and very passionate to bring back some to the country. And before joining the other field, Dr. Merchant has been associated with College of London, where he investigated various technologies for drug delivery into gastrointestinal and offered technical support and expert evidence over there. He has been working with the Abbott Labs as well. He holds a PhD and a master's in pharmacy from University of Karachi and his PhD from University College London. PhD in quality management. So I think with all his vast experience, it happened that I had the honor of listening to him and his videos as well. Quite a composed, mature man for his age. So over to you, Dr. Merchant.
Well, thank you very much for the introduction and the invitation to come over to this forum and present on the vaccines. Thank you very much for all the participants who have come in today to listen to the talks. If I could share my screen, please, so I could load my slides. Brilliant. So can you all see my screen? Can you see my screen and hear me?
Yes, we can. Thank you so much.
That's brilliant. So I'm going to talk about vaccines today. I've been asked to talk about the vaccines, different formulations, how do they work, and their efficacy, leading to their safety issues, and particularly the toxic vaccines coming in from Pakistan directly or through COVAX initiatives. So just an overview before moving on to COVID vaccine. If you look at normally how vaccines do work, we do have quite a lot of types of vaccines historically in use across the world. So we do have live attenuated vaccine, particularly we have got the nasal flu vaccine, which has been used in the United Kingdom for all school-going age children, and the oral polio vaccine, which has been used in Pakistan particularly. One of the two key examples of live attenuated viral vaccines, if you like. Inactivated vaccines, we have got some examples around is hepatitis, which is a very popular vaccine containing inactivated virus. Also the injectable polio vaccine and the flu vaccine is also inactivated viral vaccine. The next part of the vaccine is subunit or recombinant vaccines, where you have got a particular part or antigenic portion of the pathogenic organisms, which has been used as an immunogenic ingredient to get the immune response. And there are different examples of these kind of vaccines already in the market. Often these vaccines are also conjugated to provide more immune response in the patient's body. We also have got toxoid vaccines. These are particularly important for organisms where the pathogenicity is caused by the toxin released by the organism. So the vaccine target the toxin instead of the organism itself. And diphtheria and tetanus is one of the examples of the toxoid vaccines, which we have already. And then a newer type of vaccines, which has been developed in the world more recently is the genetic vaccine, which involves the viral vector vaccines and the DNA or mRNA-based vaccines, which I'm going to talk in a bit more detail from the COVID perspective as well. So when we look at the COVID vaccines in particular, then we have genetic vaccines, which are very popular, were the front runner and came out in the market first. So we have two different types of genetic vaccines with regards to COVID-19. The first one is the messenger RNA-based vaccines. The second one is viral vector vaccines. Both are genetic vaccines in which the genetic material is being administered into the body to produce an immune response. In mRNA-based vaccine, genetic material is taken into an artificial lipid envelope, whereas in the viral vector vaccine, a virus vector is used to carry the gene you need to produce the immunogenic response. The conventional vaccines for COVID-19 comes again in different categories. The most common one is whole inactivated virus, which is a conventional technique being used for various other vaccinations in the past. And some of the speakers have been talking about their ease of manufacture and how quickly you could bring a vaccine to the market. The second type is a split virion, where you break the virus particle down into pieces. And the third one is purified parts of the virus, which could provide immunogenic response. So in particular for COVID-19, spike protein is one of the target for these kind of vaccines. So parts of the virus historically been purified by breaking down the HL virus grown on a culture media, but this has also been done as a recombinant engineering, where the proteins have been produced by biotechnology rather than being taken from the HL virus. So going to the genetic vaccines first, genetic vaccines are, although they are new and people are concerned about they're being developed fairly quickly, but the history goes back to 1980s, the early times where we were struggling with some genetic disorders. And this strategy has been used for gene delivery of for conditions which are very rare, and the genetic disorders where the people were suffering and there was no other way to fix them except to repair the gene or correct the faulty gene. So to deliver the genes, viral vector has been used historically in the clinical trials, and they have a bit like checkered past after 1999 incident, where these viral vectors produced some antibody enhancement effect in some trials participants, and some deaths among the trial subjects made these kind of delivery systems very unpopular. That led to a lot more research into non-viral vectors to deliver the genetic code, and world was trying to move away from the viral vectors for the safety concerns. But in parallel, there have been quite a lot of research being put into viral vectors and trying to develop viral vectors which would be less immunogenic in terms of body's hyperimmune response to prevent the antibody enhancement effect, and try to silence those viral vectors so that when they go inside the human body, they don't have a capacity to multiply or grow inside the human body, which was one of the key reasons for the severe adversities with the viral vector vaccines. But at the same time, non-viral vector becomes more popular. But the issue with the non-viral vector was that how efficient they would be to deliver the genetic material inside the cell. So there is about 20-25 years research going on into both sides of technology to design something which would mimic like a vesicle and the cell signaling pathways would allow that vesicle enter to the cells and produce an immunogenic response. Some of the vaccines which is already being produced or are being produced even before COVID-19 using this technology was Ebola virus, Zika virus, dengue virus, HIV, and then COVID-19. So this technology is being exploited for the dangerous pathogens of today already. And while we were so busy in the pandemic, Merck got approval for its Ebola virus vaccine last year at the end of 2019, This vaccine used exactly the same technology which they have then used, the other companies have also used to produce COVID-19 vaccine. So this was the first genetic vaccine, if you like, which worked on the basis of a viral vector. The second one came quite recently in 2020 summer, when we were so busy talking about COVID, and Johnson & Johnson and Janssen got the EU approval for their Ebola vaccine. And that was one of the big breakthrough to get this out, using the vectors which the company was also trying to use for their COVID-19 vaccine. And this gave them a precedence of success and safety record to move faster on this domain. So how does the mRNA vaccines work? So you take the spike protein from a coronavirus and try to learn its genetic code. Genetic codes in terms of messenger RNA contains the instructions like a cooking recipe, and a cell's biomachinery can then use this genetic code to produce a specific protein. And then you encapsulate this genetic code into a lipid coating to make a vesicle which is then able to pass through body's barriers once injected into the system and go inside the cell to transfer the genetic code inside the cell, where the actual biomanufacturing process is happening, where the cells are producing proteins on a regular basis on the command of the natural DNA or the genetic code present inside those cells. So you would like to bring this genetic material inside your own body cell, like a cooking recipe, so your cells can produce this particular spike protein. So you're not cooking the whole virus, but that little bit of spike protein on the top of the virus, which mainly attaches to the receptors in the airways and causes the disease. So you package this molecules, artificially prepared lipid vesicles, into the vaccine. You inject them inside the muscles. It goes into your body cells, gets the entry, goes in the cells, passes the genetic information, and then the spike protein starts to be produced. Once these spike proteins starts to be produced, these cells become abnormally looking like cells to the immune system. So your own body cells start getting infected with these COVID proteins, if you like. And that attracts an immune response, not only at the level of antibodies, but also the cellular immune response, which try to then destroy all these abnormal cells being produced as a result of this vaccination. So your body produces antibodies and also produce a T cell response, the specific type of immune cells, which is involved in killer response or phagocytosis or apoptosis kind of processes, where they destroy all these cells. So your body then are prepared to react against these COVID spike proteins. So when the actual virus comes in, body recognizes these as spike proteins as a potential threat. The whole machinery is already active. You have the cellular immune response already there. Your memory cell memorize this behavior because of the vaccination. And you've got humoral response, where you got the antibodies produced. So the virus gets neutralized if it comes into the body much quickly because of this pre-exposure process by vaccination. Some of the examples of this technology is being used in Pfizer vaccine, designed by BioNTech, and the Moderna vaccine, which has already been approved and been used in some parts of the world quite a lot. This is a typical recipe of a Pfizer vaccine, where you can see the active ingredients is the Pfizer's mRNA genetic code. And then there are lots of other active ingredients which are used to design this lipid capsid around this genetic material. Normally, if you inject a messenger RNA into your muscles, your body will destroy this external genetic code very quickly. Body has got ability to neutralize all the external genetic sequence coming into the body very, very quickly. So our defense systems are very strong, which is why we have to encapsulate it in a viral looking like particle to make sure it deceives the body's defense and the enzymes neutralizing or hydrolyzing these mRNAs, and then get into the cell. If they don't get into the cell, you will not get a response, which is why you've got these lipids and some polyethylene glycols to protect that vesicle, improve the biological half-life of those moving vesicles to allow the vaccine to go into the cells and produce response. Also, you've got some buffers and some osmotic agents to make sure the vaccine formulation is osmotically similar to the body fluids. So it doesn't cause any reaction just because of its hyper or hyposmolarity to the internal fluids. So it's compatible in other ways. The second part of the genetic vaccine, or the second type rather, of the genetic vaccines are the viral vector vaccines. These viral vector vaccines again learns the genetic code from the spike protein, and then this genetic code is then being purified and then encapsulated. Instead of a lipid vesicle, it's been encapsulated into a viral vector. For example, adenoviral vector has been very successful for other vaccines and also COVID vaccines. So it's got a bit of success. This vector has also been used for gene delivery, and there have been many generations of adenoviral vector being developed over the last 40, 50 years. So we're talking about 4th, 5th generation of adenoviral vectors. These viral vectors are being silenced now. So in the past, these viral vectors also work like a virus. So when they go inside the body, they also start multiplying and attract an immune response. But the current viral vectors being employed in COVID vaccines are being silenced. What does it mean is that the virus has lost its ability to multiply in a human host. So when this virus goes in, it doesn't multiply as a virus. So it's a bit like an inactive virus itself. So this virus goes in, this engineered virus with the genetic code of the spike protein goes inside your muscles by injection, and it starts producing your body, it goes into your cells, again like the previous vaccine I've mentioned, and then your body starts producing the spike proteins. Once the body starts producing spike proteins, your immune system reacts and try to again mop up the mass produced, destroy all the defected body cells, and also neutralize them through immune system, humoral immunity like antibodies. So again, you get both cellular immunity plus you get a very strong antibody response to neutralize these kind of viruses. But because adenovirus is also antigenic on its own, so there's a double immune response, one towards the adenoviral vector itself, and the second against the spike protein produced to get you the immunity for the COVID-19. Adenovirus is normally a virus which is found in human nose, and there are different types of adenovirus which infect human population, and they cause a very mild cold and flu-like symptoms if you get contracted with a pathogenic adenovirus. Normally, these are not very dangerous viruses, and they're normally known to the human population already. There are some adenoviruses which are used, which are found in animal body instead of human body. They're known to infect animals like chimpanzees, for example. And again, they call them mild disease, and they're not very dangerous. So the adenoviral vector used in the vaccine technology is silenced, so it cannot cause pathology. It does not have its own genetic material inside, so it cannot cause an adenoviral-based disease. But because it's a virus and body see it as a virus, so body also react against that virus and produces antibodies to neutralize this virus. So one of the examples you have got for these type of vaccines is Oxford University's invented vaccine produced by Serum Institute or AstraZeneca. We have got the CanSino Bio vaccine from China using exactly the same technology. We have got Janssen, Johnson & Johnson viral vector vaccine using exactly the same technology. And for your particular interest with regard to Pakistan, the Sputnik 5, the Russian vaccine, also uses exactly this technology. So there is an issue with the pre-existing immunity with these viral vector vaccines. And because adenovirus vectors are known to human population for a long time, so most of us in our population, we would have exposed to this virus before in our life. And that means if we had exposed to this virus before in our life, our immune system already knows that this is a pathogenic virus. Although this is an engineered virus, which looks like a real adenoviral virus, but this is not. but body looks from outside and see this virus as a adenovirus. So your immune system gets activated, your memory cell memorizes this, and it starts destroying this virus. So if the body reacts against this adenoviral vector and it starts destroying this virus, then obviously this virus would not be able to go inside your body. It would not infect your body cells, and your body cells will not produce an immune response, which you need for the COVID protection. So this is one of the challenge for the adenoviral vector vaccines, that how would you not tell your body to react against this vector because you have put it in purposely and you don't want your body to destroy this. So this slide shows you a data from a viral vector vaccine from one of the manufacturer, and this data comes from the phase 2 and phase 1 trial they did. On the top, you can see the cellular immunity. So one of the parameters is interferon gamma production by the body. So this is the real human volunteers in phase 1-2 clinical trials. You can see that in people who have pre-existing immunity, but their immunity was low for the adenoviral vector, they did respond quite well for the 3 different doses of the vaccine, and they produced good cellular immune response. But when you look at the people who were having a high neutralizing titer or very high amount of pre-existing immunity to adenoviral vector, their immune response was quite low compared to those who didn't have a very high pre-existing immunity. Now, this difference doesn't look too much in this graph, but bear in mind this y-axis is logarithmic. So you're looking at log reduction here, which is at least 100 to 10 times less than what you see in these kind of population. Also, you can see this big, huge error bars. So there's a huge variability between individuals responding to the vaccine within that group. And that could be linked to the severity of the pre-existing immunity they had and how quickly their body destroys the vaccine before it produces any immunogenic potential for COVID-19. And this is serious, and many vaccine manufacturers recognize this issue with the vaccine. In terms of looking at the population prevalence, when I looked at the clinical trial data, I can see that majority of almost 100% of the trial participants were healthy subjects, but they all had pre-existing immunity against 85, were not viral vector. But 50% of them roughly had a very high pre-existing immunity, and the remaining 50% had a low pre-existing immunity. So this could vary between populations and the environmental exposure to different geographical areas. The same behavior you can see here on the humoral response or the antibodies produced, and you can see here that this is quite a lot of reduced effects in people with pre-existing immunity. So what Oxford University did in their vaccine design to bypass this effect, they use a chimpanzee's adenoviral vector instead of from the human adenoviral vector. Again, this viral vector is silenced, so this cannot multiply in our body. It just deceives our immune system. And this virus, again, does not contain the adenoviral genetic code completely. It has been genetically modified to contain the COVID-19 spike protein producing genetic code inside. So it just acts as a vector in this vaccine. But the beauty of this design is that our body is unlikely to have pre-existing immunity against this viral vector. So AstraZeneca predicted that the immune response will be great across population groups, even in those people who are elderly and normally their immune system doesn't act like a healthy subject or an adult person. They're gonna get a good immune response. But you heard in the news that when they injected their second dose, the body was very clever and recognized the viral vector, and the memory cells remember the chimpanzee's viral vector coming in, and there was some immune response, but body wasn't aware of chimpanzee's adenoviral vector, so immune response was delayed. As a result, the efficacy was great in the first injection, but once the second injection was administered, now body was very clever. Body knew, like, oh, right, adenoviral vector coming in again from chimpanzees, and body destroyed and attacked the vaccine. So they got a bit of an issue with their vaccine efficacy in this respect. And they've been trying to work out if they reduce a half dose so the body doesn't memorize or produce a very strong immune response against the chimps adenoviral vector, that maybe the second dose could be able to produce a lot more immunity. So they're trying to play with the dose and the immune response against this pseudovirus to improve the efficacy of the vaccine in human volunteers. Another company which is using viral vector is CanSino Bio from China. CanSino Bio has got a good background information from using these viral vectors. They were formed as part of the split from a big vaccine manufacturing group in the past, so they retained that technology. And they've been working in this area for a long time to develop other vaccines and genetic delivery approaches for other diseases or the rare genetic disorders. So they had a long-standing history and background experience to do this. That's why they come up with this very quickly. They've also used their proprietary 85 genovector 5 silence vector. And they also struggle with the challenge of pre-existing immunity in their vaccination schedule, which is why their design potentially uses one vaccine in some trials to see what one dose would be enough and how long the immunity would last compared to the 2-dose response, and see how does it work. So their phase 3 trials are still awaited, and we will see how this performs. But the phase 1 and 2 data we have got available so far has got good immune response, but they do seems to have some trouble with pre-existing immunity. The third one is Janssen and Johnson Johnson technology. This is the same company who had an approval of the Ebola vaccine from regulatory authorities. And they use their patented AdVac vector technology, which is again an AD26 viral vector. This is also being silenced, and they also use their PER-C6 cell technology to grow them in a cell cultured environment. So it's quite proprietary in terms of its growing the vector, producing, propagating the vaccine, and the vector itself. So the Russian vaccine, the Sputnik 5, uses a very clever technique to overcome the pre-existing immunity issue. What they do is they use AD26 vector and AD5 vector in the 2 different doses. So what they do is when they give their first dose, they use AD26. So your body develops some immunity against the viral vector, memorizes this, but the second dose uses a different vector. So you try to deceive the body and not let the body to destroy the vaccine particles immediately because of the memory of the first injection. And this vaccine has been produced in 2 different formulations. One is GAM COVID-Vac, and the other one is GOM COVID-Vac Lyo. The first one is a frozen vaccine, which would require sub-zero freezing temperatures. And the other one is a lyophilized powdered vaccine, which would benefit from easy storage conditions to the part of Russia where they would have struggled taking the vaccine through. But the vaccine being offered and manufactured on the wider scale is the frozen vaccine because of the ease of production and the scale of production needed for Russia and outside of the world as well. But that's something interesting. And recently, last week, AstraZeneca has also started a new clinical trial in the United Kingdom where they've given the first dose of their vaccine, and instead of giving the second dose of their own vaccine, they've been trying to use AD26 vector-based Sputnik injection. So they're combining the first dose of AstraZeneca vaccine and the second dose of Sputnik AD26 vaccine to see if they're going to get a better efficacy and response by combining these 2 vaccine products in the United Kingdom. So that's been kicked in, and then soon we're going to get some news about how this works in reality in patient. We have got quite a lot of non-genetic vaccines going around the community, in different parts of the world, being trialed and also being used. The one on the top of the list is a US company called Novavax, and that vaccine is a subunit spike protein vaccine which has been encapsulated in a nanoparticle and is being adjuvanted by their own patented Matrix M adjuvant. Novavax vaccine is not a purified subunit vaccine, rather it's been manufactured using a recombinant technology where you produce these viral-like particles which only contain the spike protein, for example, but they reassemble as a nanoparticle, and it's been adjuvanted by their own patented Matrix M, which is again a 40 nanometer particle containing a saponin from a natural source mixed together with some lipids. And that adjuvation is important because compared to genetic vaccines, these vaccines do not infect your body cells. They only get an immunogenic material in there. And in some patients with a weaker immune system, with the age, for example, or the people with immunocompromised, or the people who are taking some immunosuppressant drugs, or people with HIV kind of thing background, people with transplant, they do not respond very well to these vaccines. And even the response in some healthy subject is not as great as you would expect for a genetic vaccine, where you force your body to act against it. So this Matrix M kind of adjuvants, they've been added in to wake up your immune system, to increase the immunogenicity of this co-administered spike protein into the body. And that in combination produces a great immune response. So Matrix M is not developed just for COVID. That's their proprietary adjuvant being made and being used in other technologies as well. So some good news just came earlier this morning that an efficacy trial was going on in UK, was run by Manchester, and about 15,000 participants in UK from 18 to 84 years. And at least about 27%, over 65 people were in the trial. So the company just published their news preliminary assessment of the phase 3 efficacy trial today, this morning, and the efficacy overall is being suggested as 89%, where the efficacy against the SARS-CoV-2 is 95%, and the vaccine was also found very effective against the variants, the new variants, particularly the UK variant, and also the South African variant. It was about 85% effective, and the cumulative overall efficacy was about 89%. So that was a great news, came this morning. I was reading, so I added into the slides to tell you this. And the MHRA has already started reviewing the paperwork to approve this vaccine in the UK. And UK has been partnered with Novavax to run this trial. So that's a great news. And it also provides a non-genetic alternative to the population here who might not be suitable for the genetic vaccine, or where the vaccine might be contraindicated in some subjects. Some other non-genetic vaccines in the pipeline is Valneva's vaccine, which is a whole virus inactivated vaccine. This one is a French company, and it contains dual adjuvants, the aluminum hydroxide and CPG 1018. So both are proprietary adjuvants used in this particular Valneva's products. So alum is not very proprietary. It's also available as a generic adjuvant, and many other manufacturers has been using it. But CPG 1018 is from Dynavax, and this is a very interesting adjuvant. It is a TLR9 receptor agonist, and what it does is it stimulates your B cells to produce more antibody producing cells. So when the whole inactivated virus comes in along with this adjuvant, you get a very high potentiation of antibodies. So this particular technology in the past has increased the antibody titer many folds. One of the examples of this adjuvant is already present in hepatitis B vaccine, which has been FDA approved and used in the US as well. Another interesting non-genetic vaccine, which is in the pipeline, is a Medicago biopharmaceutical company in Canada. So they have developed this COVID-19 viral-like particles, and these particles are grown in plants. And they uses a GSK's ASO3 adjuvant technology. This is again a patented proprietary adjuvant from GSK. It combines squalene with tocopherol and polysorbate 80, and that potentiates the immune response. Again, Medicago's vaccine is non-genetic. It contains a spike protein as an antigen to produce an antibody response. And in combination with adjuvants, you would expect a better immune response compared to the spike protein alone. The same adjuvants has been offered from GSK to Sanofi and Clover Pharmaceuticals in China. But recently, the results of the Sanofi were not very great. And Sanofi's COVID vaccine using the same adjuvant produced antibody response, which was quite similar to a patient who's recovered from the natural disease, like some similar titers as in convalescent plasma. The titers were not very high compared to other vaccines. The titers are many folds higher than the naturally acquired immune titers, which means you get very strong protection and you're going to last longer. So this had a setback, and the Sanofi has been reformulating and trying to improve their vaccine design to see how they could improve the results. Similarly, GSK has been working with Medicago and Clover to come up with a better produced, a better response vaccine in that aspect. Two other vaccines which are very important from Pakistani's perspective is the conventional vaccines from China. The first one is the Beijing Institute one, which has been manufactured by Sinopharm. It's again a whole inactivated virus vaccine, which has been adjuvanted with alum. So you have, again, the classic immune adjuvant in there, and has been already been used in UAE, Bahrain, and has been considered for Pakistan and soon going to be used in the population. The other Chinese vaccine, which has been in the pipeline for since a while, has been used in Indonesia, Brazil, Turkey. Singapore has recently signed an agreement to get it. It's called Sinovac. Sinovac is also inactivated whole virus, which has also been adjuvanted with alum. And many people see these two are same vaccine. They use same technology made by two different companies. But there is one key difference between those two vaccines. They use two different types of strains of coronavirus, and that could affect the population immunity and efficacy in the geographical spread, where a particular strain would produce more antibodies for a particular geographical prevalence of COVID-19 variants, for example. So they're not really same vaccine, although they use the same technology, they look alike, but there is a key difference in there, which could be very technical in terms of assessing them for a particular region or community. So I'm not going to go in detail of this one, but this is just an overview of what type of vaccines being ordered by the UK. So you can see we have got viral vaccine, viral vector vaccine, non-viral vector vaccines, and UK already ordered Novavax and GSK vaccine a long time ago. So Novavax producing promising results today means we can quickly go in to an approval phase for Novavax and then start using that one. The Valneva vaccine, which is being manufactured in a facility in United Kingdom near Scotland. It's a French company, and that vaccine has also been funded by the UK government. It's an inactivated viral vaccine, and that is going to come, I think, by the mid 2021. The phase 3 trial started in the winter, so it will take a while to come out, but that's another big non-genetic vaccine coming out in our markets. In terms of vaccine efficacy, I'm not going to spend too much time here, but if you've got questions later, you can ask. I'm running short of my allocated time here. What I would like to say is that the efficacy claims in all the vaccines, not only a particular vaccine, is being based on interim assessment of the phase 3 results, and sometimes the data is limited in interim assessment, so the real life efficacy is likely to be lower, much lower than what's been seen in the claims. So the claims are not fake, they are not false, but this is what it is in the interim assessment, but they are not real life efficacy. Also, because of the viral challenge is not possible, it's difficult to differentiate between trial subject in the placebo and trial arms. The antibody levels and the T-cell response in many phase 3 is not being known very well, although we have phase 1 and phase 2. And the PCR testing is being very limited based on symptoms. So you can't practically offer a PCR test to 44,000 participants in the vaccine on multiple time points. And that's one of the limitations where the efficacy might have been overcalculated or estimated, because some subject might be missed out and not been offered the test, particularly the asymptomatic carriers in the both groups. The life of antibodies and the future booster doses would dictate the vaccine efficacy in the long run. So this is very important. And there is another big reason that majority of the vaccine trials were done with a normal saline placebo, where a BCG control or something which provides a more generic immunity against pathogen would have been nicer to see how does vaccine differentiates from the other typical vaccines being used in some parts of the world. For example, BCG vaccination is very high in Pakistan, and that would be very interesting to see if that has got some role to play. There was a clinical trial running in Australia about BCG repurposing for COVID, and we're still waiting to see how that spin out, but that could be a good news for Pakistan coming down the line. Also, these efficacies are subject to range of genetic variants coming out from different geographies, and not just the super mutant ninjas you've heard about UK and South Africa. There are other genetic variants prevalent across the world, and this real life efficacy depends on genetic prevalence of COVID virus, how it's been changing across communities, because these vaccines are not trialed everywhere around the world for their efficacy, and particularly phase 1 and 2 were very restricted in certain parts of the world. So the translation of information or interpretation of data of efficacy is not just comparing numbers. It requires a lot more understanding of how these things are done and interpretation based on a particular part of the world. And also the pre-existing immunity, not just against adenoviral vector, as I said, but also in some parts of the world, coronaviruses has been producing local endemics and pandemics, not pandemics, but epidemics in those regions. For example, SARS-CoV-1 producing SARS, the Middle East respiratory syndrome coronavirus, you had Nipah virus before. So those pathogens would have also produced some kind of pre-existing immunity, some population, which may have some maybe different efficacy response compared to other populations, which may not have those pre-existing immunity against the viruses. So this is what we said in November 2020 when the higher efficacy claims were published and people were talking about in the press. So we published this article talking about why these efficacy claims would not be very realistic and real life efficacy will be lower. There's a link here if you wanted to read more information about that. So vaccine efficacy, again, in terms of safety, the majority of side effects are not really troublesome. I'm really running out of time, so I'm going to skip this fast. But if you had got questions, please feel free to ask me later. So anaphylaxis is again very rare, but the incident rates are higher than what we normally see with the flu vaccine. Then reporting vaccine side effects, there is a parallel scheme across UK, same in Canada and US, to report the side effects from the vaccine directly by the public healthcare professionals, and anyone can use that. And this is the MHRA-based system for the UK. But also DRAP in Pakistan has partnered with MHRA and WHO and come up with a very efficient safety reporting system, which is app-based, and it doesn't really require an active internet connection. By the time you're reporting, whenever your phone will get access to internet, it will transmit the data later. So it's a very good platform to collect the pharmacovigilance data once the vaccination kicks in Pakistan. So that's very important that we really sign up to this kind of platforms to collect. So some of the known safety issues, which has been, for example, this is officially coming from Pfizer. recommendations for the vaccine recipients in the UK. So this has been given to all healthcare professionals. So potentially the allergy, hyperallergic responses to the vaccine is one of the contraindications. Also, if you were allergic to any other vaccine components, or you got breathing problems in the past, or you currently have an active illness, then you should not take this vaccine without your doctor's advice. If your immune system is very weak, for example, because of HIV or any other conditions, for example, organ transplant or the drugs you're taking, then again, do not take this vaccine without your doctor's discussion. If you have a bleeding problem or bruise easily, or have been using some anticoagulants like warfarin or DRAP, some new direct-acting anticoagulants, then you should discuss with your doctor before you take the vaccine. Some other warnings and precautions is kids under 16, we haven't got the safety data, pregnant women, breastfeeding mothers, and then also some ability to drive. So when you get the vaccination, you've been advised not to drive immediately or see if your ability to drive has been affected for a little while. This is not a very common effect, but it's found in some cases, in some rare cases. So there's a caution that you do not immediately drive or use heavy machines. There are other potential risk groups which I really would like to mention for the genetic vaccine. So if you remember the mechanism of action as I explained with the viral vector vaccine or the mRNA-based vaccine like the Pfizer or Moderna vaccine, your body does produce immunity because your cells start producing this COVID vaccine. So your body will attack this adenovirus itself and then produce an immune response, and your body will also attack these defected cells which are now producing the antibody. So these are your own body cells which are being infected by this genetic material. So in the normal circumstances, in majority of people, this will be perfect, and your immune system will not only destroy these defected cells, but they will also produce lots of antibodies and cellular immunity to keep for the coronavirus, for the future immunity. But people who have got immunocompromised situation, they might struggle clearing this mess up. So there's a bit of caution that they discuss with their doctors before they go and sign up for any viral vector or genetic mRNA-based vaccines, and maybe some other vaccines like poly-inactivated viral vaccines or Novavax kind of technology may be more safer for those kind of patients. Similarly, people with past HIV or in the current phase of HIV or who are in the convalescent phase, all those kind of people, even who are recovered from HIV, normally do not properly recover from HIV. So those people also should be cautioned about these kind of things because they do have a history of deficient immune system, and you might have a long-lasting or a long-run safety implication for these genetic vaccines. And currently, we don't have any dangerous effect to really get scared of, but because the safety data is very limited, and these effects are likely to be seen on a longer term. So there's a caution here for some population, particularly other people who have got hyperimmune response, who react with a hypersensitivity to vaccine ingredients like anaphylaxis or even non-anaphylaxis hyperimmune response. They might be likely to react with viral vaccine because there's a history of overreaction in the past with these viral vectors. So people with severe allergies, people with cancer particularly have got some defective cellular immunity because your body's immune response cannot kill the abnormal looking like cells in your own body. So if you had a past history of cancer or a current cancer, then you had some kind of indication that you've got some indication that your body might not be able to recover from this process. So one more group I would like to mention is people with thrombocytopenia in the past. So people who would have got dengue virus infection in the past might also be careful taking a genetic type of vaccine, and speak to their doctors, speak to their consultants, take advice before you move on and take these kind of particular vaccines. So COVID variants are common. They're not something of new. There are different types of genetic mutations we have seen, and we have got different type of these mutants being formed over time. We have got type 6 variants being more dominant in the last of 2020, and then the type 7 being emerged from the UK particularly, which we have seen. So COVID genetic variation will be common, it would not be surprising. And this was come out very recently, where in an in vitro study, what they've done is they've challenged the virus into a selective pressure in the presence of convalescent plasma containing a very high and effective titer of neutralizing antibodies for the virus. And they found in 45 days, some genetic mutation happens in this challenge test. And then after 80 days, they've got 3 distinct mutations in the virus, which makes viral variant completely resistant to neutralization against those antibodies. But this is done in an in vitro setting. This has been really challenging the virus from high selective pressure. So this normally would not happen that quickly in a human population. But this is an objective evidence in combination with in silico prediction, where you could now work out how long the virus will then mutate and get to that mutant stage where the antibodies will not neutralize them. So it's important that we look at other therapeutics options, not just rely on one side of therapeutics, and we look at the antiviral drugs particularly. I think I've got only 3 minutes left in my talk, I think. So, or I've already gone over. So how much time I've got, Major General?
Ahmed, the time is definitely over, but definitely we would love that you conclude with this fascinating presentation.
Thank you. I'll try to sum up very quickly. So a couple of antiviral drugs which has been ignored, particularly in Pakistan. And I would say this is something we need to put some efforts on. We've done some good clinical trials in Pakistan, and favipiravir could be a promising candidate. It's an oral drug, and lots of people who are dying with the disease, they can't have a vaccine. They're already dying in ICUs and they have got severe disease. They need something to neutralize the virus now. And these kind of antiviral trials would still benefit the society. So I think the Pakistan should try to get this one in. This one is owned by one of the Japanese companies who has already offered this product to any country who wanted to run it for the trial free of cost. So that's a potential opportunity to take this in. This has already been used in Turkey and many other countries as a first protocol for COVID-19 in the first week to neutralize the virus. There is another interesting antiviral drug called molnupiravir. This is also a very broad spectrum drug. Again, an oral drug which can be really spinned off very fast, very quickly, and shown very good efficacy in animal trials. And it has suppressed the virus spread in ferrets within 24-48 hours. But again, this came out from a US university, a small university in the US, but they didn't get the sponsor in time. And a very small pharma company now has funded their phase 3 trials. They just kicked in their phase 3 trials in December. And there is an opportunity that Pakistan can work with these guys and bring a clinical trial for Pakistan, which is not just a trial. You're going to give a hope to all those people struggling with COVID without having any, neutralizing antiviral drug. So that's another thing I would say. Some precautions about social distancing, I'm going to skip. But early intervention therapeutics, I would like to touch on that just for 1 minute, saying if you understand how the virus goes into your nasopharynx, causes loss of smell, goes down to your mouth, causes your loss of taste, goes down to your oropharynx, produces the symptoms of cough, and then comes down to your laryngopharynx, goes to the lung, comes down to the stomach, causes gut infection, goes to the liver, causes all sorts of problems. So the early intervention therapeutics, which we proposed early on, works on nasopharyngeal sanitization concept, which really uses some very simple things like steam with subessential oils, antiseptic mouthwashes, which has shown efficacy against coronavirus, antiseptic throat rinse, which can reduce the viral load in your throat and your mouth, some anti-inflammatory throat rinse, which can reduce the cytokine release, inflammatory markers directly at the source. You can use some antiseptic lozenges, which can stay in your mouth and slowly release the double antiseptic ingredient from your throat down and get more coverage in the laryngopharynx as well. And some immune support in terms of vitamin C, D, and E. All these products are all licensed products available in the European market. Majority of them available in Pakistan as well. And it's already been, some of the products are already being licensed by EMA for use against viral pharyngitis. So there is an opportunity to use this in Pakistan to provide first-line defense against the virus. So anybody who get the virus should start on this strategy immediately to get their chances of getting immunity as quick as possible. I'm going to skip all that. There are lots of evidence, but if you wanted to know more and ask me questions, I can talk about that in details. But skipping that, at the end, I would really want to leave some messages for Pakistan that COVID vaccines are not just COVID vaccines. And Pakistan has to really leap into biopharmaceutical manufacture. We need to consider manufacturing COVID vaccine now, then later. We need to do a tech transfer, need to do TTO or whatever is required. Pakistan need to move on. It's not only about COVID vaccine, it's about future security, future safety of the country, and also about the medicines for tomorrow. We need to invest in some antiviral therapeutics in Pakistan, as I mentioned, a couple of drugs, which will be very interesting to bring in. And also, we need to work out pre-existing immunity in our community against 85 and 8026. Without having that idea, it would be really difficult to understand which kind of viral vector would work better in our community. We need to do seroprevalence studies in Pakistan. We know we can't screen everybody in the country, but a representative, a population representative covering from north to south, a statistical sample would give us some idea about how much pre-existing immunity is in our community, and are those immune titers are neutralizing the prevalent strains in our countries. So genetic surveillance is really very important, and many vaccines has been trialed in Pakistan. Some vaccines are coming in, and what we can do is we can do the seroneutralization assays in vitro. NIH is the brilliant place. They've got the capabilities, and we can find out which vaccines are working in our population, which vaccines are neutralizing the genetic variants in Pakistan, and predict the future variants in Pakistan, so we can have a booster and a future manufacture in Pakistan according to our own needs. And also, I would suggest this early intervention trial, the oropharyngeal sanitization. This concept should be implemented nationally in Pakistan. This is very cheap, easy, doesn't cost a lot of money, very safe. We are not giving any wrong medication. They are already the formulation produced for your oral rinses and mouths. We are not repurposing anything. It's all there, all regulatory approved, all approved by EMA, European Medicine Agency. Why not we use it in the population? We all can manufacture it locally in Pakistan. The products are there, and this would provide a lot of support to our population who can recover from the infection naturally until the vaccine comes in and we acquire the vaccine, acquire immunity. At the end, I've got just lots of acknowledgements to make, and thank to the CPRDC, NIH, and GMS, in particular, Major General Ahmed Saab and Dr. Jose for the invitation to come and speak at that event. Thank you very much.
Thank you so much, Merchant. That was really amazing. You're doing a fascinating job, and we need people like you back in Pakistan, or maybe from there, whatever. And however you can help, I mean, it would be a great thing. As we move on to the question, I would like to share this my own feeling. I think one person that has been missing from the talks has been Dr. Uzair, who I think should be given an additional degree of PhD in social networking for providing the platform, sitting in Canada, I mean, putting the people in UK, Canada, France, what and what not. I mean, he just has the contacts and putting. Dr. Uzair, I would like to you to say a few words because you've got an amazing experience with vaccines in Pakistan, and just a couple of minutes, if you can, please. Over to you, Dr. Uzair.
Thank you very much to everybody. Assalamu alaikum. So thank you very much, particularly Amir Ikram, to give me a very precious few minutes. So because I am in love with vaccine for the last 25 years, so that's my passion. It's not just the knowledge. So I try to connect the people who have massive knowledge. So I just attended a seminar, a webinar by Dr. Hamid Merchant. I realized there are so simple measures that we can bring to Pakistan. There are so simple treatment which can be available to Pakistan. So we are going to bridge the knowledge gap between the developed countries and the developing countries. So I'm really grateful to my very valuable speaker who spared their time, particularly to Dr. Zulfikar Bhutta. I think he has the other overlapping opportunities. He canceled some of the meeting. So because, and you know, there is a lot of messaging coming up looking for the future seminar, because this is an extremely knowledgeable. I was just responding to a lot of questions. You know, all the people, they are asking, please, please, please have more such series of seminar. So I will request, you know, the General Amir Ikram and the Hamid Merchant, please do consider some future latest development, at least for during the emergency time, maybe once in a month. I will request if you can spare time. I'll be really grateful. Thank you to all of you. Thank you.
And thank you so much, Dr. Zaid, and thank you, whatever you're doing, sitting far and away for the country. And again, I'm sure you're coming down in February, that would be the time. So many questions have been asked, some of them a bit gloomy, pessimistic. No, the picture is not that grim. The topic is even light at the end of there is a light at the end of the tunnel, and we need to concentrate and be very watchful in our near future. Just very briefly, I would like to say that some of you are asked, NIH basically is the sole producer in the public sector. There are few in the private sector. Whatever is around 7% of the country's requirement is being fulfilled locally, and rest is all being imported. So we have, I think, a huge challenge in front of us. Maidan khali hai. I would like to say this word in Urdu as well, that we have plenty of opportunities. A huge number of studies have been conducted, 6 in total for Pakistan, starting from 93, 97, 2003, 2016. And unfortunately, every study went on the carpet with WHO, World Bank, UNICEF, so many other parties coming into it. 3 years earlier, once I joined NIH, what we did, we took out all the studies. Frankly speaking, I had gone through all of them myself, prepared a crux, and now is the time for public-private or a totally private venture. So we have taken this responsibility. We prepared a model for the government, which has been approved now, and we would be rather invited before COVID, all the parties which are interested in producing vaccine within the country, because we have a huge demand for even EPI alone, which is the expanded program of immunization. So we are trying to provide this platform to the private sector. Even during this crisis, we have done the mapping. If you get the bulk, who's going to take care of that besides NIH? At NIH, definitely all the units which were shut down previously, just to mention like measles, 7 years, for 7 years it was closed, and we immediately rejuvenated it once I joined. It is producing measles vaccine and so many other units that we have rejuvenated, and we have produced, recently inducted a new CGMP qualified anti-CPRDC production plant, which can fulfill the country's requirement. So some of the questions that get answered from my comments over all these. But coming down, Merchant, there's a huge number of questions on. So I am sorry, I'm overwhelmed by the questions from the audience. Extremely grateful to you because of your passion and total concentration during, and I have been following your comments even during the presentations, how keenly you are following. Many questions. If we'll be able to answer a few, but we have all the questions and we will send it to all the experts for their domains. They will reply back and we'll definitely share over the emails. But many questions about messenger RNA vaccines, how long they remain active and effective, and how long they continue producing the spike protein in the body. Over to you, Merchant.
It's a very interesting question, and as you could see that the mRNA goes into your body, starts infecting your body cells, and then your immune system reacts. So ideally, in an ideal world, in a healthy individual, you would achieve that in a few weeks, and then your body would stop producing those antibodies or the spike protein antigens, if you like. And that's because you are injecting a fixed amount of load into your body. And once that load has been neutralized by your cellular immunity, is gone. That's where the risk comes in for some population where the immune system doesn't respond properly. The people who are immunocompromised, has got HIV, have got history with other conditions, or taking some drugs which suppresses immune system, or been going through this kidney transplant or other organ transplant. In those people, we might think that the production of those spike proteins may go on for long, and that messenger RNA wouldn't stop. How would you stop it? Which is why we have got options here that people who are in those clinically contraindicated groups, where these particular type of genetic vaccines may not be very safe than the other vaccine, then you can have that discussion with your doctor, with your consultant, and see what your medical history is like. And only a healthcare professional who knows your full medical history can give you a personalized advice as to which particular vaccine is more safer and appropriate for you. So luckily, we are in a world where we have got different technologies, and we have got non-genetic vaccines. We have got the whole inactivated viral vaccines, where the virus will not be active and actively producing and causing any disease. So if people are immunocompromised, they can also make some immunity rather than having no immunity at all. And there's no risk of being infected from the vaccine. Similarly, you've got Novavax vaccine, which is in recombinant technology, but also provides immunity in the same pattern. So it's a bit longer answer to your question, but I've tried to explain you the reason behind, so you do not just get scared from the vaccine and make a kind of more judicial choice.
Dr. Bhutta, I'm bothering you. There have been so many questions asked about the booster dose, even the second dose, why the second dose is required, and what is the possibility, how long the immunity would remain in the body, and what is the possibility of a third booster as well. Over to Dr. Bhutta, please.
So the honest answer to many of these questions is we don't know. We don't know. This is a new virus. And if it behaves anything like the influenza virus, there may not be long-term immunity. So there are lots of these things that we're going to have to look at. But I think, as was pointed out, some of the T-cell mediated immunity data are very encouraging. And also the reality is that despite all the noise and millions of cases, over 100 million cases worldwide, cases of reinfections within the first 6 months or around that time after the initial illness are very, very rare. So there is enough hope that the immunity against this virus can be protective up to a year from what we recognize, and hopefully for longer, depending upon how these new technologies stimulate your immune memory cells, T cells. From the data available from some of these vaccine studies, and Moderna has it, Sputnik vaccine has some very good T-cell mediated response data, AstraZeneca. All of these seem to be stimulating immune memory. So the answer to the question is we are confident that there may be immune boosting. Sputnik vaccine relies on the principle of prime boosting, which means that you prime with one and you boost with another. And they're doing that. And AstraZeneca and Sputnik are joining hands with another trial like that. So a lot of this information will emerge. Now, today, we have for the first time the one vaccine, the one dose vaccine results released by Johnson & Johnson. I've just been looking at those this morning, and they have an overall protection against severe disease of 85%, but only about 60-some% efficacy against COVID-19 infections per se. So we may have to come back to this idea that a single dose vaccine of this may not be as protective as a double boost. Now, the big global challenge, John Amr, as all of us will have to face, is that we are very rapidly going to get into a situation of multiple choices for COVID vaccines, having a hierarchy of vaccines with some that have been shown in studies in certain geographies of over 90-95% protection, and others less so. And then we will have to very scientifically look at the comparison of vaccine impact in one region of the world versus another. So I don't think it's fair to compare vaccines that have only been studied in, for example, North American populations, which are not very diverse, versus North American diverse populations, those that include elderly versus those that do not include elderly subjects, with vaccines that have been truly tried in low and middle income countries. So lots of eyes are on studies and trials that have actually been done in Asia and Africa and parts of the Middle East to see what is the response in the right kind of population. Just to close, you must have seen how Germany, for example, yesterday took a decision, their regulatory committee took a decision of not recommending AstraZeneca vaccine for people above 65 years of age. And that's a very hard decision because they could not convince themselves that there was sufficient data in that age group. I think it's going to be very challenging to find adequate data across all age groups, across all vaccines, because they've been designed very differently for emergency use. And we will have to some extent, rely on immunogenicity data with age groups like that as efficacy surrogate. So I think it's a bit of an unfair decision by Germany, but it's something that we need to keep in mind. Thank you.
If I could add a couple of points to this,
sure.
So I would completely agree with what Dr. Bhutta has been saying, and it's absolutely important to bring that into the equation. And the phase 1 and phase 2 trials shows you that evidence that how a elderly subject reacts, whether they produce antibodies or not, and that's a very good predictor in the absence of the real phase 3 efficacy data in the age population. But in terms of booster doses, just to add a bit more on that, yes, you will need booster, and you might need booster adjusted according to how the virus is changing. And the technologies to produce vaccines today allow us to change the vaccine recipe very quickly and come up with a newer booster regime, which could provide you extended protection against the variation. If I try to link it back with the flu virus, if you remember the Spanish flu pandemic, we in UK at least seen 3 waves of Spanish flu with 3 distinct mortality curves, and then the pandemic died down about into this time. But is H1N1, H3N2 left us? Never left us. It comes every year in the winter season, kills thousands of patients, and we try to protect them with an annual flu immunization program. Currently, we have a quadruplant vaccine, particularly an adjuvanted trivalent vaccine for elderly 65 plus or 85 plus people. But we do that every year to protect the most vulnerable in the society. So booster doses would be common, and there might be a possibility that coronavirus will stay with us forever, like a flu virus, which has never left us. It's part of our ecosystem now. Millions and millions of people have now been infected with the virus. The virus can't leave us. It will only adapt to our human host. But some people will be at risk, elderly people, immunocompromised every year, and we might need to protect them. And there is a possibility that maybe in quadrivalent flu vaccine, we add a 5th ingredient in there and maybe have a combined flu immunization with COVID immunization. That's highly likely for a long-term protection over the years. So there is a light at the end of the tunnel. Whatever we are trying to, kind of warn in this, webinar today is for the better scientific understanding and better planning. But there is definitely light at the end of the tunnel. It doesn't look very gloomy. There is a solution to this, and there is a long-term solution for the long-term problem. But we need to focus on the interim issue now, but in parallel, we need to press all those buttons to make sure we are protective for the future. Thank you.
Thank you so much. I take this opportunity, I feel privileged to have such experts on the panel today. We've definitely crossed our time as well. Once again, I would like to, just to concluding the session, I would like to say that extremely grateful to the Council General in Canada and the CPRDC in particular, because they're very much keen, and I think it's time to bring back, as I already said, things back to the country. To all the experts, we knew Dr. Bhutta was committed, but very deeply indebted as he has been there throughout. Hamid, Irfan, Obed, Hamid Sahab, Hamid Sahab, everyone, and starting from our organizer, Zafar from the Global Services, and all his team. I think starting from Ms. Sidra to all others who have been there. And definitely, last but not the least, Mr. Uzair. I think he has been brainchild behind this. There have been huge requirement of such, as you already told. We'll try to grab in much more opportunities. And for those who are asking for Dr. Merchant's presentation, it would be great, Hamid, if you can send your YouTube link so that they can even watch all those live presentations. That would be good. And once again, thank you to all of you and very, very dear participants who are still there from the beginning. And let's hope we meet very often. And my message is just to conclude, it's all, we have to join hands in order to sail. There is definitely a light at the end of the tunnel. Let's grab it and fetch this opportunity. Thank you so much. Assalamu alaikum ji. Bye-bye.
Welcome, Salam.
Walaikum assalam, and thank you very much indeed to all the panelists, organizers, and all the audience who have been very patient and listening to us. Thank you very much.
Thank you to everybody and goodbye.
Everybody.
Thank you, everyone. Thank you so much for your participation, and thank you to all the speakers and all the panelists for joining in. Thank you so much.
Thank you.